researchers revive dead organs. 9 out of 10 patients in need of organ transplants wait in vain for healthy organs. However, researchers have now managed to revive organs in dead bodies using a blue liquid. This breakthrough could potentially save the lives of thousands on waiting lists, and one day even help living patients. The process must happen swiftly, within hours after a person's death. Organs like hearts, lungs, and livers must be transplanted into waiting patients. Within just a few minutes without oxygen, cells and organs begin to die. The organs only receive fresh oxygenated blood once transplanted into recipients, which stops further deterioration. If organs completely die, there is no turning back. This, however, may no longer be the case. A research team from Yale University in the United States has cracked the code for reviving dead organs and keeping them alive for valuable extra hours. Their method involves pumping a cold blue fluid into the body, which rescues cells in dying organs using oxygen and minerals, allowing the organs to continue functioning. After several successful trials on pigs, the researchers are now working to adapt the technique for humans. Organs die quickly. Organs rely on a constant blood supply. When death occurs and fresh blood stops circulating, cells no longer receive oxygen and nutrients, and they can't expel waste. As a result, cells swell, their membranes burst, and they die. So far, the most effective way to preserve organs has been to cool them, as lower temperatures slow down biological processes. In 2021, 20 people died each day while waiting in vain for an organ transplant, according to data from the Council of Europe. Cooling only delays the inevitable, though. The most resilient organs, like kidneys, can survive for up to 36 hours before transplantation. Hearts and lungs, however, can only survive for 6 hours outside the body. These tight time limits make it challenging to meet the demand for healthy organs. As a result, only about 10% of patients on global waiting lists receive the organs they need. A global shortage of organs. The world is currently experiencing an acute shortage of healthy organs, according to the latest figures from the Global Observatory on Donation and Transplantation (T), led by the World Health Organization (WHO). The total number of organ transplants performed annually is around 144,000, including 92,532 kidneys, 34,694 livers, 8,409 hearts, 6,470 lungs, 2,025 pancreases, and 172 intestines, which only meets about 10% of the need. The most common method for keeping organs viable today is the use of a mechanical lung known as ECMO, extracorporeal membrane oxygenation. ECMO is a pump system connected to a living patient, where blood is enriched with oxygen and recirculated into the body. This method can keep patients with heart and lung failure alive while waiting for healthy organs, but ECMO cannot maintain organs after a patient has died. Fresh oxygen saves dying cells. In their new experiment, the Yale research team developed a fluid capable of keeping organs alive for significantly longer periods. The fluid, which contains 13 different nourishing and healing substances, is mixed with blood and can partially replace it in the body. The fluid is enriched with oxygen and includes synthetic hemoglobin, a protein naturally found in red blood cells, which binds oxygen from the lungs and releases it to the rest of the body's cells. The researchers also add essential minerals and anti-inflammatory substances to the fluid, which inhibit cytokines, signaling molecules that usually call immune cells to remove dead cells and repair damaged ones. In a dead body, the task of removing dead cells becomes a major challenge. Cytokines attract more and more immune cells, which eventually do more harm than good. In 2019, the research group published striking results from experiments treating pig brains with the fluid. The brains were remarkably kept alive and performed normal functions for up to 4 hours after the pigs had died. The team has since further developed the concept, now called OrganEx, using a pump system. They tested it in a series of new experiments on pigs that had suffered cardiac arrest induced by electric shocks. An hour after death, half of the pigs were connected to OrganEx, while the other half were treated with ECMO. After 6 hours, the organs of the pigs treated with ECMO had deteriorated significantly, while those treated with OrganEx were in excellent condition. The organs had activated genes that promoted self-repair. Cells in both hearts and kidneys absorbed glucose from the OrganEx fluid. Researchers also detected electrical activity and rhythmic muscle contractions in the hearts, which achieved normal heart rhythms and healthy blood pressure. In both the liver and brain, cells began producing new proteins necessary for organ function. According to the research team, it was difficult to distinguish the organs of the dead pigs from those in living pigs, even under a microscope. Blue fluid revives organs. In several experiments with dead pigs, a research team at Yale managed to revive organs that had suffered heart attacks. Using a cold blue liquid, the organs began to repair themselves and resume normal functions. The researchers behind OrganEx see great potential not only in alleviating the global organ shortage, but also in the possibility of treating organ failure in living patients, or even potentially reviving a patient after cardiac arrest in the future. Our research demonstrates the body's ability to resurrect after death, says David Andreevich, one of the researchers behind OrganEx. The method raises profound questions among experts about where to draw the line between life and death. Some discuss whether the pigs whose brains were revived using BrainX could be considered conscious. The researchers themselves say no, since there was no coordinated electrical activity in the brains. However, other scientists caution that we are entering an unexplored gray zone with both ethical and legal implications. OrganEx, to an even greater degree, challenges us to rethink our understanding of death, explains David Andreevich on Yale's website. Not all cells die immediately. It's a prolonged process. It's a process that can be interrupted. The debate among experts now revolves around how much of a body needs to be revived before we can say that an entire animal, or eventually a human being, is truly alive again.